dosing, timing, and which individuals might benefit most from this strategy. Closely tied to the story of NAD+ are the sirtuins, a family of 7 proteins that act as critical regulators of cellular health and longevity. You can think of sirtuins as the CEOs or managers of the cell. They don't actually perform the labor themselves, but they oversee and direct a wide range of essential processes, including DNA repair, inflammation control, metabolic regulation, and circadian rhythm. Their activity, as we just learned, is entirely dependent on the availability of NAD+. When NAD+ levels are high, sirtuins are active, and the cell is in a state of high alert, efficiently managing stress and maintaining itself. The discovery of sirtuins and their role in aging came from studies in simple organisms like yeast. Scientists found that an extra copy of a gene called SIR2 could dramatically extend the lifespan of yeast cells. This discovery was really groundbreaking because it showed that lifespan could be modulated by a single gene. Researchers soon found the equivalent of these longevity genes in more complex organisms, including humans. Our sirtuins, particularly SIRT1, SIRT3, and SIRT6, perform analogous functions, playing a pivotal role in protecting us from age-related decline. The activity of sirtuins is naturally increased during times of energy stress, such as caloric restriction or fasting. This is because these conditions increase the ratio of NAD+ to its reduced form, NADH, effectively making more NAD+ available to fuel sirtuin activity. This is actually one of the key mechanisms by which fasting and caloric restriction exert their pro-longevity effects. This understanding led to an intense search for compounds that could directly activate sirtuins pharmacologically, essentially mimicking the benefits of caloric restriction without the need to restrict calories. This search led to the identification of several sirtuin-activating compounds, or STACs. The most famous of these is resveratrol, a polyphenol found in the skin of red grapes, blueberries, and peanuts. In the early 2000s, resveratrol generated massive excitement when studies showed it could activate SIRT1 and extend the lifespan of yeast, worms, and fish. It was also shown to improve the health and survival of mice on a high-fat diet, protecting them from metabolic disease. The idea that a compound from red wine could be a key to longevity was understandably very appealing. However, the story of resveratrol in humans has been more complex and frankly, kind of disappointing. While it shows clear benefits in animal models, the results in human clinical trials have been largely inconsistent and underwhelming. One major issue is bioavailability. Resveratrol is poorly absorbed and rapidly metabolized in the human body, meaning it's difficult to achieve the high concentrations used in lab studies. While the principle of activating sirtuins remains a highly valid and promising strategy for promoting longevity, resveratrol itself may not be the magic bullet